You can have generalized transduction. Generalized transduction then happens anywhere in the DNA sequence. It doesn't really matter. It's just going to insert itself somewhere. Specialized transduction the virus or phage inserts itself in a specific spot in the DNA sequence. This is useful because then we can target specific genes if we want in our effort to move them from one cell to another, using viruses as that vector, if you will. Last type of gene transfer we're going to talk about, conjugation. Conjugation is what happens when bacteria transfer genetic material from one cell to another cell. And it's really as close as we're going to get to bacterial sex. Basically, you have one cell. There's its genetic material. But the cell also has a plasmid. This particular plasmid gives this cell the ability to transfer this plasmid to other cells. Conjugation requires contact with other bacteria cells. But if we have another bacteria cell that comes in close proximity, there's its genetic material. What can happen is a pili from this microbe connects with that microbe. So this is through pili. And in fact, it's a specific pilus called the sex pilus. Those cells that have this plasmid are said to be F plus cells. They have the fertility plasmid. Those cells that lack it, then of course, are S minus. They lack the fertility plasmid. A F plus cell will only connect up with an F minus cell. You don't get F plus cells connecting up with F plus cells. What happens is that this sequence of DNA codes for the sex pilus and codes for the enzymes necessary to allow for it to move after it's copied itself from this cell to this cell. So now this cell becomes a fertility plus cell. So the F plus plasmid is a self-transmissible plasmid. It can code for the enzymes it needs to make the sex pilus, to copy itself, and to be transferred. Not all plasmids can do that. Some plasmids use the F plus plasmid and piggyback along in their efforts to get transmitted to other cells. So we'll see the F plus plasmid being a self-transmissible plasmid, but also being used, if you will, by other plasmids to get moved to other cells. Yes?
So the F minus cells reproduced by any which other types? How do you get how do you get another F minus cell? Because if the plasma gets transferred.
Well, you mean, you have to realize there's probably millions of F minus cells out there. And so not every F minus cell is being converted. And so that F minus cell, when it replicates into two F minus cells, they replicate into two F minus cells.
By using binary fission?
Yeah,
Oh.
yeah. So when this replicates by binary fission, the plasmid will replicate with it. And so the daughter cells will have the plasmid as well.
Can you explain one more time how the F plus cell, F minus cell plus?
Okay, so this is an F plus cell because it has the plasmid. This is an F minus cell because it lacks the plasmid. The genes of this plasmid code for the sex pilus, so it codes for this. And also the genes code for the enzymes that allow for the plasmid to be copied and to be transmitted. Okay, so when contact is made with an F minus cell, and it has to be an F minus cell, then the plasmid, using the genes that allow for it to transmit itself, forms the sex pilus and then moves across through the sex pilus into the new cell. And so now this cell here has a plasmid.
So basically the transferation of plasmid from F plus to F minus is making F minus to F plus.
Yeah, it's converting this now to an F plus cell. A couple of things that we can say about plasmids. They're found in most bacteria and archaea, and even some eukaryotes, but that's not as common. They often play a key role in the functioning of the cell. What I mean by this is a lot of times the plasmids will impart to the cell some characteristic, a new protein, a new enzyme, allowing it to take advantage of a new fuel source or allowing it to produce an exotoxin. In most cases we're talking about, or I shouldn't say in most cases, in all cases we're talking about a double-stranded DNA sequence. It's circular. The number of genes can vary. Some plasmids only have 4 genes, and so they're not going to be self-transmissible. They need help. Other plasmids may have as many as 1,000 genes, so it really can vary depending on the plasmid.
Does the F plus cell become an F minus cell after that, or does it still retain a plasmid?
No, it retains the plasmid because the plasmid copies itself before it transmits it. Yeah, so it retains it, stays in the F plus cell. And then plasmids, for the most part, have a very narrow range of cells that they'll infect. So one plasmid may not infect all bacteria cells, only a specific species or a specific type. That's not to say that there aren't some that do have a wide range, because there are, of course, going to be exceptions to everything. Okay, any questions? All right, so next time we meet, we're going to do a little bit more genetics. We're going to talk about Richard Linsky's study, and then we'll do some review for the exam on Monday.